Mendel was doing his work in the 1800s, really the mid to around 1870, I think is when he died. So at that time, Mendel was really working in the dark. He was understanding inheritance, but he didn't know what the inheritance was really based on. For example, he didn't know what DNA was. He didn't know that chromosomes were the genetic material, but he figured out a lot of stuff about inheritance still. And then what later scientists did was they linked what Mendel saw with what they knew about chromosomes. And that's really how we get the chromosome theory of inheritance. It's really connecting what we saw with Mendel's work to what we now know about chromosomes. So this looks really intimidating, but it's basically things that you already know, and we're just going to link them back to what Mendel saw. So first of all, chromosomes contain the genetic material, and that genetic material is passed from parent to offspring and from cell to cell. And also remember that genes are within the chromosomes, so you can have hundreds of genes on the same chromosome. Okay, the second piece is that chromosomes are replicated and passed from parent to offspring, and that during replication, each chromosome is copied individually and divided individually. And again, remember, you know this because you've studied mitosis and meiosis. So remember, each chromosome gets copied separately and then will be lining up on the metaphase plate and then passed on to the gametes. The third part is that the nucleus of a diploid cell contains two sets of chromosomes. So again, remember that diploid means that they have homologous pairs. And that one member of each pair comes from the mother and the other from the father. And when we talk about homologs, they're virtually identical. It's very, very small changes in the chromosomes that lead to the traits that we have. And so this, remember, aligns to what Mendel saw, which he said, okay, you have flower color, but each individual has two alleles for flower color. Whoops. Talking about flower color, so I should be using P's, right? So each individual has two because they have a homologous pair of chromosomes. So again, Mendel didn't know it was chromosomes that he was talking about, but that is what was happening. And then at meiosis, each chromosome pair separates, and so that during that separation, each chromosome pair separates independently. So again, remembering what happens on the metaphase plate, the homologous pairs line up independently. Each homologous pair is separate from other ones. And so that's where the law of independent assortment and the law of segregation are going to come from. And then the fifth part is just about fertilization. So gametes are haploid. They only have one set. They'll have to combine to form a diploid cell during fertilization, which is then how we get back to having two sets of chromosomes. So again, this is all mostly about mitosis and meiosis and what we already know. So let's just take a couple of more closer look at segregation and independent assortment. So again, remember that we have homologous pairs of chromosomes. So one copy that's inherited from the father is a paternal copy, and one that's inherited from the mother, the maternal copy. All along those chromosomes, you have genes. And the homologous pairs are functionally equivalent. They have the same information on them. So for example, in this position, on the both members of the homologous pair, we have the gene for plant height. And you can either have a tall allele, or the plant can have a tall allele or a dwarf allele for plant height in this location. In this case, this is a heterozygote, so they have a copy of the tall allele and a copy of the dwarf allele in that homologous pair. So during meiosis, remember the first thing that happens is this will get copied, so now we have sister chromatids, so two copies of the tall allele and two of the dwarf allele. Meiosis 1 will separate the homologous pair, so now I have two haploid cells, but the sister chromatids are still together. So the second division, so meiosis 2 now, will create four haploid cells where the chromatids are now separated, and each one has either a small t allele or a big T allele. So Mendel saw that you saw separation of the alleles so that each gamete only had one allele. And again, that's because of what happens to the chromosomes during meiosis. So the behavior of chromosomes also explains the law of independent assortment, and this has to do with what happens on the metaphase plate. So if we look here, now we're following the Y and R genes for the seed color and seed shape alleles, and we're looking at a plant that is a heterozygote. And so this is the OR. This is the same plant, right, starting with the heterozygote. So on the metaphase plate, you could get the chromosomes lining up to where all the red ones are all on one side, or you can get it where the blue and red is on each side. So this is the metaphase plate. And again, see how the red is on one side here, but blue and red is on this side here. And so if they line up like this, then you'll get in this case, you'll get the big R with the little y. So this is going to give you gametes like this or like this. And if they line up this way, you're going to get gametes that are like this or this. So notice that's all four of our gamete combinations. The little y with the big R, the big Y with the little r, the big Y with the big R, and the little y with the little r. So really, both of these are happening to get you all four possible gamete combinations. And again, that's why we see independent assortment rather than linkage.